So as you know, there is now a lot of initiatives to personalize cancer therapy according to the genomic alteration. And there are two main reasons for creating this conference. First, the patients are being offered some genomic tests that are now available, and there is no education for the oncologist to interpret the result of these tests. So the first reason why we wanted to create this conference was to educate and share some way of interpreting the genomic data from the patients. The second reason why we have created this conference is to have some consensus about how to interpret the genomic alteration. Example, there are different trials across the world trying to show the clinical utility of doing genomic testing, but each of these different trials use a different interpretation tool of the genomic data. So one of the goals is to standardize and to better understand how can we interpret the genomic data. So in some way, personalized therapy is already used in cancer. For example, we give ALK inhibitors according to ALK translocation, or we give HER2 inhibitors when the patient presents HER2 amplification. So what we are preparing now is the next step, is to what extent using or screening for several hundred genes can improve the outcome. And for this, the trials are ongoing, but we are facing some issues that are the fact that we don't know really how to rank each genomic alteration when we screen large number of genes. So what are the next steps in this field? The next steps are clearly to better rank each genomic alteration and to better understand the clinical meaning of each genomic alteration. When we will be able to do that, then we will develop a second generation of trials that really will show clinical utility of this kind of approach. I think in the future, blood tests will be part of the molecular analysis, but personally I think it's unlikely that blood tests will summarize the whole portrait of the tumor for many reasons. First, because until now it's very difficult to assess protein activation using a blood test. Second is because we know that we need to personalize therapy according to the activation of immune system, and until now most of the predictors are being developed in the tumor, so we need to assess the immune system in the tumor. And also because the next generation of biotechnology like metabolomics or other things like this will be applied in the tumor. So personally I think that blood tests will be a very important tool, but it's going to be only one component of the whole portrait that will include biopsy of the cancer.